So my name is Laura Privalle. I work for Bayer CropScience, and I guess I have the pleasure of being the first representative from industry to give a talk at this meeting. And I'd very much like to thank the organizers for the invitation. And I have to say, I think this is such a timely topic, and I'm very much looking forward to the rest of the outcomes. So my topic is, what does it take to bring an ag biotech seed product to market? So we're going to go a little bit behind the scenes and paint the picture of what goes on. We're going to talk about the process for bringing an ag biotech product to market, possibilities and realities. What have we learned? Originally I had said failures there, but when I went through review, they made me change that to what have we learned. And then we'll have a summary. Okay, so we've already discussed this a couple times already this afternoon, but this is always where I like to start my presentation. And on the top, there's traditional plant breeding, where the idea is that there's a desired gene, that's the orange bead, from the donor plant that's sexually compatible with your commercial variety. When you make a cross, you bring in the desired gene, but you also bring in a bunch of other stuff that you might not want. In plant biotech, the desired gene can be from any donor. It does not have to be sexually compatible. But the only gene that's brought over is the desired gene. This part is not regulated. This part is regulated. So I always like to start there and sort of reset and get back to that point. Okay, so what do you do when you want to bring a biotech product to market? First, you have to have the idea. Then you have to identify the gene or the trait that allows you to impart that idea into the crop of interest. You have to be able to transform the plant, the crop plant, and usually one single cell that then has to be able to regenerate into a whole plant that is actually fertile, that has the trait of interest, functional. You have to confirm the functionality, usually in greenhouse and then finally out in the field. And then you have to select the event that's going to make you a bunch of money. That, in other words, the market is desirous of having and is willing to pay you some added value in your seed product. Does this work all the time? No, it doesn't. What can go wrong? The idea can be bad. It just doesn't work. It's too complex, perhaps. Maybe the gene is not expressed. The gene can be inserted into a poor location. We've had a lot of talks about that already today, and that's really the basis of this whole meeting. By a poor location, it could harm the plant, so the plant cell may never survive tissue culture. You kind of have a good hint that that's the problem when you don't get any regenerates back. You can impact plant metabolism, the phenotype. You can get albino plants. You can get non-fertile plants. You can get weird stuff like this. This is tassel ears. So when you first bring a plant through tissue culture back to the greenhouse, it really, especially if it's corn, it pretty much looks like crap. But if you're lucky enough for that plant to make a seed and for it to be fertile and to pass the trait on to its progeny, you get normal-looking plants in future generations. It's also possible that the trait is not inherited in a Mendelian fashion. And by that it means it's not incorporated into the nuclear genome. It could be into the chloroplast or the mitochondria, so you have not normal segregation, which makes it very hard for the breeders that are going to be producing the seed that you want to try to sell. On the other hand, maybe it could be a desirable place to have it transformed into the pollen. That's a whole different story. But one of the questions we ask is it inherited in a standard Mendelian fashion? And you could have yield drag. Not too many growers are going to be interested in a crop that doesn't yield as well as their comparative crop. That wasn't the case for Roundup Ready soy originally. It actually had about a 5% yield drag, but it was of such value to the grower that they didn't care. And then eventually it was bred into varieties where that yield drag didn't happen. Okay, so this is a numbers game. You have to start big. You have to have many ideas to get the right one. You have to transform many cells and get lots of events. Then you have to evaluate all those events to find the guy that's going to really be worth the investment to bring to market and worth somebody buying from you. Okay, so this is the results of the Phillips McDougal survey that was published, I think that was in 2012 now. And it depicts the idea that you start with a whole bunch, you go down to your single event, then you introgress it and bring it to market. Couple things about this. You're starting with huge numbers. So this is mean units. This could be genes, ideas, events, all combined into one to eventually get to, usually you get one and a backup and you commercialize one of them. The other important point is that there are lots of months involved. I think when I converted this to years, it's 15 to 16 years. Now, when I first started in the business, which was many years ago, they gave us 10 years to get a product. That's how long the company was worth investing in biotech. And if it didn't happen then, they may not have supported us anymore. Fortunately, at that time, it took us 10 years to get something into the regulatory system, and so there was a glimmer, hope at the end of the tunnel. And by 12 years, we had a product out. And those were back in the days when we didn't know how to transform corn. We didn't know about having to codon optimize your genes. We didn't know how to get single cells turning back into plants, regeneration of corn, all that kind of stuff. Those problems were solved, and it took us 12 years to get that first product to market. Now it takes a lot longer, and this is really the big difference. The last phase, getting approvals, global approvals around the world before you can actually launch your product. Okay, so many ideas never make it past the discovery stage, and hence the regulators never see. I and several of my colleagues here, we were in the Philippines giving a presentation to regulators and non-regulators several years ago. You remember, Greg? And they came to me and they said, how come you never bring something that has problems? Why does everything get approved? And that's because, actually that's the point of this talk here, to demonstrate that we go through a lot of sifting to identify the guide that's not going to have problems and be able to bring that. So 10 to 15% don't pass the allergen toxin homology screen. This is a very conservative test, or the criteria are very conservative, and so you lose a lot of your possible traits at this step. Now, certainly, as we all know in the world, there are not 10 to 15% of all proteins out there are allergens. So a lot of the good stuff goes out with maybe one or two bads, or much fewer, I should say. It's possible it's not expressed in the plant. It wasn't efficacious. You know, sometimes the plant comes back and bites you in the butt, and that's kind of, you know, I kind of like that. I sort of root for the plant once in a while, so just to prove we don't know everything. It's also possible that we're actually able to generate a product scientifically, but the market doesn't support it. There are lots of crops out there that don't have enough value to support the cost of bringing something to market. Okay, so only the best performers with the correct attributes get promoted. And here's the same point. Economics can play a role. And as I said before, some products with good solutions can't support the cost. Is this any different from conventional breeding? Typically, when I had a conversation with one of our breeders to get this information, they typically do 80 to 100 crosses, though some may do as many as 600, to try to identify new products or try to trace down new traits. It's pretty similar for all crops. And using contra-season, so growing in more generations than just one through the year, it can take 6 to 7 years to develop a new cotton, soy, or wheat variety. And marker-assisted breeding can actually help in bringing multiple traits together at one time. And I really respect those guys, and your brain has to work a special way to be one of them. Chemical mutagenesis, as was discussed earlier, is also useful for identifying or producing simple and useful traits. In that case, the development can even take a little bit longer than the 6 to 7 years. And biotech traits are incorporated using forward breeding, or they can be presented as populations in a trait integration project. So there is breeding that goes on the tail end of a biotech product development, depending on the kind of crop that's involved. Okay, so how do we go about selecting the elite biotech event? Number one, I always tell the research folks, is efficacy. It's got to do what you want it to do at a level that, is worthwhile. So it's got to be formed. It's got to perform. That's number one. It can't impact agronomic performance. As Phil said earlier, I'm not sure a duck equals a duck, but a soybean plant's got to look like a soybean plant, act like a soybean plant, grow like a soybean plant, except for that attribute that you've introduced. So it can impact yield or other agronomic parameters, and it has to have the correct inheritance pattern. Okay, those to me are just obvious and extremely reasonable, because you want to produce something high quality that your grower, your customer, is going to, want to buy from you. But the molecular characteristics of the crop also are a very big driving set of criteria for selecting one event from another event. It should be a single locus, should have a single copy of the cassette inserted, so you could have more than one at the same locus. I'm saying you should have one copy, one locus. Should lack vector backbone, should only have the promoter, coding region, terminator cassette that you're interested in. It should retain its integrity. So it should be the complete. Actually, for good performance, it probably needs to be complete. But sometimes you'll get ones that don't have quite all the promoter, or don't have quite all the terminator, and that might be okay. But if it's complete and you have a choice of 5 events and one's complete and one's not, you would go for the complete. Has to have stability across generations, and you should know something about the insertion site, the organization of the locus. Okay, how important is the insertion site? This should say play a role in the efficacy of the trait. It's important for designing your event-specific tests. Knowing a lot about the insertion site can be impacted by whatever crop you're working with and the amount of genome information that's available. So these are some of the main crops for which there's a genome sequenced. But as Wayne pointed out, just because that's your sequence in your Williams variety does not mean that that's your sequence in your Brazilian variety or whichever line you used for transformation. There can be differences, there can be SNPs, there can be, just if everything was the same, we wouldn't have variety. So you can be tripped up a little bit by how much is known as well, because then you have to justify, why is this one nucleotide any different? It's different. The insertion site is very important in the EU, Japan, and other places. Not so much, I think, to the U.S. regulators or to Canadian regulators. And if I'm wrong, you guys can yell at me. What's important is to not have interrupted a gene, to understand the organization of the locus. Now, all of this stuff ignores the plant performance. And to me, if the plant didn't care, that's the most important thing. Again, does it look like corn, taste like corn, yield like corn, have the same compositional components as corn? Then why does it matter actually where the insertion site was? Okay, so what I'm saying is it ignores the plasticity of the plant, and occasionally it can lead you to overinterpretation. You don't know, but if the plant performs, the plant didn't care. There can be varietal differences. I mean, just because that just reflects differences in the inherent genome. Okay, so the modern events are produced using agrotransformation and have fewer insertion issues. I threw this in because the genome of soybean was published, and I highlighted this point. It's highly duplicated, with nearly 75% of its genes present in multiple copies. So actually, if you landed in one of those genes and the plant still performs well, then it's compensated. It probably didn't impact anything. Okay, so the earliest events were generated using biolistics, kind of brute force. This is the so-called gene gun. Current protoplast transformation was also a route that was sometimes taken. BT11 was produced through protoplast transformation. Today, we take advantage of the very elegant agrobacterium tumefacient system, where it's a lot more precise, and you can dictate better what goes in. Now, sometimes doesn't always happen perfectly. That's again why you do many transformations and screen many events. Okay, so once the event is identified, then the safety assessment is conducted. And I just really enjoyed the presentation by Phil McDougal earlier today, because he emphasized this point, that what we're trying to do is we're trying to show that the genetically optimized plant is as safe as the conventionally bred plant. And we consider human health and environmental safety. Okay, so it's a multi-pronged approach. We look at the safety of the crop and its performance. We look at the safety of the gene and the protein. Products produced through biotechnology are the most studied food products. They're reviewed by agencies all over the world. Last year in the survey, the ISAA survey, 29 different countries actually grow biotech crops. And a heck of a lot more countries review them, review our dossiers, because they import them. Now, Europe, I understand, is actually the world's largest importer of food products. And it has a very stringent regulatory system with the least acceptance shown by its people. This is a comparison of approvals, and I had a higher number than what Phil McDougal said earlier today, so I guess it depends on your source and how you count them. But this includes both biotech products and novel plants that might have been produced through mutagenesis or like the Clearfield varieties. So you can see that around the world, different agencies have different numbers. Now, some of these numbers are kind of inflated because some regulatory agencies around the world consider singles and stacks, whereas in the US and Canada, only singles. Stacks are not reevaluated when they're combined through breeding. So that's one of the reasons why Japan has a much higher number. There's also some data around on what's pending. Not necessarily, I wouldn't say that these are really hard numbers, but this is just what I have been able to gather. So the number of crops that are involved also varies quite a lot around the world. Okay, so what have we learned? So this was the slide that had originally said failures. So there are examples of products or ideas, they may not have gone beyond the ideas, that were stopped before approvals were sought. The Brazil nut protein is the one that's cited all the time because they were trying to improve the levels of methionine and cysteine in soybean, but the protein they identified to pull in turned out to be the allergen from Brazil nut. So that project was stopped well before it was even thought of taken to commercialization. The bean amylase situation in Australia, I think you all probably are well aware of that. That never went commercial. There are many vegetable examples, and here I put little dollar signs if the reason why those were stopped may well have been due to economics. Not because they didn't perform, not because you couldn't actually identify great events, but because the economics did not support that product. This would be increased carotenoids in tomatoes, for example. The amylose potato is another example. That was the idea, to increase the percent of amylose in the starch of the potato. That was an example where the starch levels were not high enough to meet the minimum of what was required for starch potatoes, so it didn't make it, even though you could really increase the levels of amylose in those potatoes. Now, there are also examples of events that have been taken all the way through approval and then have dropped. High lysine maize is an example. Trifid flax here in Canada was approved. It's not commercialized. Amflora potato is a sad example. In my opinion, I spent a lot of time. Christina's going to talk about this later. She spent even more time working on that product. New leaf potato. This is BT potato with yellow dwarf virus resistance, I think. That was brought to market, but suppliers for McDonald's potatoes said they didn't want to have to use that potato, so if those guys don't accept it, that's kind of history for a potato product. BT corn was the first BT maize product to be approved and was actually approved for cultivation also in Europe. It was replaced due to superior products coming along. Okay, so once approved, consumers may never see the product, or not for long. This would be the Flavor Saver tomato. The demand was high, but the product was never profitable. It was transformed into a germplasm background that did not make it worth growing, is basically the bottom line. So I tasted this tomato way back in the day. We made salsa out of it and served it on BT corn chips. I know Ray remembers that. Yeah, that was a lot of fun, but this product is no longer on the market, and neither was that maize product. So this kind of little trip down memory lane, this was the flyer for Amflora's starch potato. This was the brand for BT 176. This shows Trifid flax. This is how BT potato, which performed beautifully, even though it was made by a competitor, it performed great, but I think it was, is it McCann's or McCain's? They're the ones that said no, so but to this day it would be a great product. Okay, so in summary, it takes a lot of effort to produce the single elite event that we're going to take commercial. This is not unlike plant breeding. Evaluation of plant performance eliminates events in which the insertion site was detrimental, not unlike plant breeding. Thus, the regulators never see these guys that have an impact. The structure of the insert is an important factor in elite event selection, and characterization of the insert is a very critical element of every dossier. So reviewers around the world evaluate all of these safety packages. Again, I want to emphasize these are the most highly studied, evaluated foods or feeds that are consumed. Many products fail, but these are due to issues not associated with safety. And I want to emphasize that no product has been withdrawn or not approved due to an unintended effect related to the insertion into the genome. That's it. I'll be glad to answer any questions. Thank you.